Our question for this project was, will different wavelengths of light affect the speed of chlorella algae growth? We picked this project as we had heard how the Cawthorne Institute was testing algae cultures for environmental research, and in school, we had been working with lights in our tech classes, and we were familiar with soldering, wiring, and LEDs. Our experiment required preparation for the algae and lights before running the experiment. The algae and food were diluted with distilled water and added equally to all 6 jars. Each jar was aerated from the same pump with 6 valves to keep the oxygen content equal between the jars. We soldered the wires to the lights, then mounted them on metal brackets, and these we then mounted to the 6 boxes. We drilled holes through the metal and wood to let the wires exit the back of the boxes. We then lined the 6 boxes with reflective paper, and we separated each box to prevent light contamination. For the duration of the test, all boxes were kept in the same room to keep the temperature and humidity constant for all boxes. The room was equipped with a heater to minimize temperature fluctuation. We were then ready to carry out our test. We measured the light transmittance through the algae every 3 to 5 days. We were then able to use the light transmittance to measure the algae growth, as higher algae concentration means less light is able to pass through the algae culture and reach the intensity meter. Therefore, lower lux would mean more algae. Our measurement stopped on the 14th day. Our results are listed here. We found 730 nanometer red appeared to be the best for algae growth. Second was 515 nanometer green, and third was 625 nanometer red. The 460 nanometer blue, 616 nanometer red, and the 425 UV appeared to be the worst for algae growth. And, on our last test, all had higher lux, meaning loss of algae concentration. This could be the result of unhealthy lights or algae death from other uncontrolled variables. We would like to repeat the experiment to verify our results. We would like to test more wavelengths within each color range to find more precisely which will stimulate more growth. With this information, we could potentially grow bigger algae cultures faster, making research using algae more efficient. This would be helpful for existing research using algae to generate electricity and clean up waterways.